So till now we have studied two laws of inheritance given by Mendel. One is law of dominance and second is law of segregation. Out of these two laws, law of segregation has no exceptions. It is correct for all the examples which were taken. So there are no exceptions. But the law of dominance, when it was tested in various other organisms, it had certain exceptions. So we will be studying about the exceptions in law of dominance. So we will be basically focusing on incomplete dominance and codominance. These are the two exceptions. But along with these two exceptions, we will also study two other concepts which came along when it was studied. One was multiple alleles, that is multiple allelism, and second is pleiotropy. So let's begin with the first exception, that is incomplete dominance. Let's see it through animation first. Now let us study about incomplete dominance. Till now we have studied about the concept of dominance. But what is incomplete dominance? Incomplete means partial. So it's a type of partial dominance. What does it mean? Partial dominance means the dominant trait is not able to mask the recessive trait completely in F1 generation. So the F1 hybrids, they show intermediate characters. This type of dominance is also called as semi-dominance. Let us study it with the help of few examples. First one is 4 o'clock plant, which is called Mirabilis jalapa. In this plant there are red colored flowers which are dominant over white colored flower. When they are crossed, the F1 generation all are pink color. This means the red is not able to mask the white character, white trait. So as a result, the F1 generation shows the intermediate trait. You can also study this with the help of another example, that is called snapdragon flower or dog flower. Here also there are red colored flower which are dominant over white, and when they are crossed they give all F1 hybrids which are pink. But when these F1 hybrids, they are selved, they give the same genotypic ratio of 1:2:1. So let us study it in more detail with the help of cross. Now let us study the concept of incomplete dominance with the help of the cross, that is monohybrid cross. You can see in the case of Mirabilis jalapa plant, red colored flowers are dominant over white color flowers. So as per Mendel's law of inheritance, all the F1 flowers should be red in color. But here in this case, the F1 hybrids are all pink in color, because though red is dominant over white, but it is not able to mask the effect of white completely. So as a result, there is pink color effect. Okay. But when these pink color flowers are selved, then F2, you can see in F2, the genotypic and phenotypic ratio becomes same. Whereas in case of law of dominance, the phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. But here, in this case, both phenotypic as well as genotypic ratios are same. Here you can see capital R, red 1, capital R, small r, pink 2, and small r, white 1. So this is an exception. This is a deviation from Mendel's law of dominance. This can be seen in other organisms also. in animals also. Especially in case of Aleutian fowl, this is a kind of a hen. Here, there are two types of the birds. One are black birds, which are dominant, and white birds, which are recessive. When these black and white, they are crossed, then F1 hybrids are all steel grey in color. So this is also a kind of incomplete dominance. Now let's switch over to codominance. What is codominance? Let's find out. Now let's study the concept of codominance. The word codominance is made up of co, which means mutual or together, whereas the dominance is the concept where a particular allele is able to mask the effect of other recessive allele. So, in the concept of dominance, a particular phenotype is able to express itself in hybrid state. But what happens in the concept of codominance? Let's find out. So this codominance is a phenomena in which both the alleles are dominant, and they are dominant and express themselves together in F1 hybrid. Here, for example, in this case of a fowl, the black colored as well as white colored feathers are equally dominant, and their cross results into checkered feather. Let's find out how does it take place. Now this black colored fowl or black feathers are because of the genotype capital B, whereas white feathers are because of the genotype capital W. When this black and white, they are crossed in F1 generation, the genotype is capital B and capital W, and the phenotype is all the organisms, all these birds, they are having checkered feather. So it is neither black nor white, both, all checkered feathers. And when these checkered feathers bird are crossed, they show the similar genotypic ratio as in the case of incomplete dominance, that is 1:2:1. So both genotypic and phenotypic ratio are just similar as in case of incomplete dominance. So this concept of dominance is also shown by many other animals, like in case of cattle, that is roan, which are having the patches of red and white, because this white as well as red both are dominant. Now let's study the concept of codominance as well as another phenomena that is called as multiple allelism in human blood group. You are aware, we generally humans have 4 basic types of blood groups, that is A, B, AB and O. And these blood groups are controlled not by 2 alleles but 3 types of alleles. These are IA, IB and small I. These I represents for immunoglobulin. Our blood group is because of a type of immunoglobulin present on the surface of our RBC. If my blood group is A, then my RBCs will have immunoglobulin A. If my blood group is B, then the immunoglobulin B will be there on the surface of the RBCs. A person whose blood group is AB will have both the types of immunoglobulins, because IA and IB both are codominant. So they will appear both on the surface of the RBCs. But a person having blood group O doesn't have any immunoglobulin because he has got a recessive gene, small I. for the immunoglobulin. So no immunoglobulin is produced and such persons have blood group O. So let's see. A person having blood group A can have two combinations. He may be homozygous IA, or the person may be heterozygous for blood group A, that is IA and small I. Likewise, a person having blood group B again can have two types of combination, either homozygous IB or IB or small I heterozygous. But the person having blood group AB will have only one combination, that is IB. Likewise, a person having blood group O will have again only one combination, small I. Okay, so let's take this example. Suppose a person having blood group A marries a person having blood group B. So this person, suppose, is heterozygous for blood group A, then there will be two types of gametes produced, IA and small I. Likewise, in this case also there will be two types of gametes produced, IB and small I. And if you see the Punnett square, four combinations can be produced. What are these four combinations? IB, the person will have blood group AB. If IBI, then blood group B. Here, blood group A, and when both small I are there, then the person will have blood group O. So, this concept explains the codominance and multiple allelism. Okay, so these exceptions were not observed by Mendel.